Oh, and then I'll share my screen. Okay. Okay, so yeah, thank you guys all so much for coming to hear about how you could get involved in research at McGill or outside of McGill altogether. So just to give you our agenda. So first I'll talk about what is the McGill Scientific Writing Initiative, which is the initiative that's running this event right now. And then me, Ellie, I'm going to talk a bit about my perspective about research, and then Lorena and Alexa will do the same, and then we'll have a Q&A session at the end. So a bit about the McGill Scientific Writing Initiative. We're a club, student run, but we also have a faculty advisory board held of amazing McGill professors who help us out, could answer any questions we have. But in essence, we have workshops such as this one to help students get comfortable in research, learn about science, learn good study methods. We have a blog and we have a website with resources as well. All of that could be accessed by our Instagram, so definitely go check that out if you haven't already. And yeah, I could tell you a bit about more about MSWI at the end if you're curious, but let's get into what you guys are here for. So to start off, I'm starting us off. So yeah, my name's Ellie, and I'm in my third year of anatomy and cell biology here at McGill. And I'm from Montreal, so I did CGIP first. So then I'm really just in my third year at McGill. So yeah, I'm going to talk about how to contact labs and an example email that I've sent to a primary investigator, which is the one in charge of a lab, and then a bit about the volunteering that I do in a research lab. So, this information that I'm going to share with you in the next two slides, I got mostly from a workshop that I actually went to, held by the Anatomy and Cell Biology department, by I think it was the advisor of our department. So these were really helpful. I used them when I contacted lab professors. So that's why I'm going to share with you guys because it really helped me out. So to start off, you might think, okay, looking at one researcher that I'm curious in, I'll just email them, see what happens. You might be really disappointed when they don't answer you back because they're all super busy. So I really recommend finding 5 to 10 people that you're interested in. Let's say if you don't even know what topic you want to research in, just search up McGill Research, McGill your program, so McGill Anatomy and Cell Biology supervisors, and you're going to get a bunch of people with a bunch of little descriptions of their research. And so you could kind of get a feel from that, and then you could really deep dive. When you see one professor, they'll link another professor and so forth. So you could really explore a lot of people in any field that you're interested in. And then I would recommend finding 5 to 10 that you're like, okay, I would love to work with them. And you could read a bit about their current research projects. This you could find on PubMed if you search up the professor and see if it interests you because likely that is the type of thing that you would help out with. And then now into the emailing part. So if you want to email a professor, I recommend keeping it short, 2 to 3 paragraphs. But you can see my email, it wasn't even that short because it's hard to kind of send your message across in a few sentences. So, just try to keep it as short as you can. And try to be clear about what you want from them. So let's say if you want a work experience, say that. If you want to just volunteer a couple of hours, say that right at the beginning so that you're on the same page as them. And if you have any research experience, go out and tell them that. They'll love to hear it and it'll give them confidence in recruiting you. But as a new university student, totally fine if you don't have any. So don't be discouraged. This is how you're going to get experience by emailing professors and getting to know them. And right, why you want to join their lab. I'm going to show you an example of how I did that. It doesn't have to be a whole elaborate, I researched your lab work and I liked XYZ super scientific. I really just said I worked in the summer at a camp and I liked working with kids. And like, I think that's good enough because you're going to be, just a genuine person with genuine interests. And then attaching your transcript and CV, I think is also good. It shows them a bit more about you. And they also said in this workshop that if they don't respond in 7 days, you can definitely email back, but there's a chance that they just won't answer you. But even I think one of the labs that I reached out to, they only messaged me back like 2 months later, but they messaged me back with the opportunity that I'm doing right now. So you never know what's going to happen. Okay, so this is an example of an email that I sent. I sent out and they did message me back. I didn't end up working with them because just the time didn't work out, but I think this email could work if you want to take it and make it your own. So at the beginning, be polite, dear Dr. XYZ. And then I said, I hope this email finds you well, said my name, my program. And then I started with why I want to join that. Well, what I want from their lab. By the way, you could take a screenshot of this, and also we're going to post a recording with the slides. So you can have this for reference if you want. Um, no worries. So yeah, I said what I want from their lab. So I said I'm open to volunteering in the summer because I knew that I wouldn't have too much time to dedicate. So I didn't feel like it's worth it to ask for money. I felt like I'd be open to volunteering. So I really just wanted anything they could give me. And then I said my research experience. So I said I did research in the summer. And I also said, if you guys are familiar with Biol 301, it's a lab class, 8 hours a week in the lab. So that's really valuable if you did something like that. And why I'm interested, so this is exactly what I said, working at a camp for kids and adults with special needs, because this lab is about mental health and mental illnesses. So I worked with kids with mental illnesses, and that interests me in the topic, and it's not necessarily my interest isn't from research, it's just from common life experience, and I think that's just as valuable. And then I said my CV and transcript was attached. So I hope this email was clear, and you can definitely ask questions at the end. And then trying to sorry, the slide's not moving. One sec. Okay. Ooh. Okay, sorry about that. Okay. So then the lab that I am volunteering at now. So I volunteered 2 summers ago at a lab at UDM, which is what I mentioned in the email. But the lab that I'm volunteering at right now is a McGill affiliated lab, and it's called the Centre for Precision Psychiatry. So it's in the psychiatry department. It's the Allen Memorial Hospital. I didn't know it existed until I started volunteering there, but it's actually up the hill past McIntyre, so it's really up there. And I go in a couple hours a week and I basically copy data from a paper onto the computer. So it's really not that, you don't need much skill for it, but it gets you in the lab, and I still learn a lot by interacting with other people there. And you still learn like what you're reading is information about people's mental illnesses, mental health issues. So I'm still learning a lot. But it's just a nice way if you don't have too much time during the semester, but you still want some experience. So I definitely recommend doing something like that if you're nervous about time management. Couple hours a week, and it's just fun to be there with other undergrads, med students, MDs. So it's a really cool thing to do. And the topic of the research or our mission is to make a data bank, a biobank for all sorts of doctors, PIs around the world. They could access this data. It's known as like an open access. So anyone around can access the data and use it for their research. And the goal is to help psychiatrists and pharmacists develop new diagnostics for mental illnesses and develop better drugs. So yeah, it's really cool. Um, and definitely a fun experience. If you're interested in looking into it, you can definitely ask me questions about it afterwards, or look up the Dr. Simon Ducharme and message to see if they're looking um, for more undergrads. So yeah, that's a bit about the lab that I'm working on. And next, I'll go to Lorena.
All right. Hi, everyone. My name is Lorena, and I'm a U2 physiology honor student. Okay, next slide. So first, oh wait, I think the slide's being a little slow. Um, okay, so what I'll be talking about is first like a summary of the lab that I'm working in and the research focus. I'll be talking about my volunteering experience, which is like how I started, and then my experience as a paid researcher in the lab. And then currently I'm doing a 396 project. So I'll also be talking to you guys about that. And then I'll end with some research tips for you all. Wait, Ellie, I think you skipped a slide, like before the what I'll be talking about. Yeah, there we go.
Yeah,
So
perfect.
first,
Sorry.
actually, I'll be talking. No, you're good. You're good. First, I'll be talking about why join a lab in general. So first, you get a lot of hands-on experience. So you'll be able to apply what you've learned in class to real-world research, and you'll be able to like bridge the gap between theory and practice. For me, at least, because I'm in a wet lab, so I learned a lot of techniques like pipetting, electrophoresis, PCR, and you'll be able to learn similar techniques that will prepare you for independent research in the future. Also, you'll be able to expand your knowledge. So for me, I've learned a lot about how to read scientific papers, how to analyze data, and how to really just understand how discoveries are made in the scientific field. As well, the research community is amazing. I love my lab. Everyone in there is super nice. And I just really like working with a team of passionate scientists. As well, for me at least, I get a lot of freedom to conduct my own experiments. After learning the techniques that I needed to be more independent, my professors were like, okay, you can just go ahead and use the cells however you want, do whatever you want, use the drugs. So it really gives you a lot of creative freedom to do what you want. As well, like I said before, you get a strong network of mentors and peers. I met a lot of other professors in different fields through my lab, so you'll also be able to do that. And then I also met a lot of like-minded students who are also working in the lab, which is pretty cool. And then last but not least, you can discover your own interests and determine what aspects of research excite you most. And it's a valuable self-discovery process that'll help guide your future career path for sure. Okay, next slide. Okay, so I'm working in Dr. Alvin Schreier and Dr. Gil Bubb's labs. I don't know if you know them. They taught Fiji 209. I don't know how many guys are in physiology, but yeah, so their area of research is cardiovascular physiology, and specifically, they talk about cardiac dynamics. So Schreier's lab is focused on ion channel function, localization, and the role in the initiation and conduction of cardiac rhythms. So they use a lot of techniques to try to understand that. And also in his lab, he uses mathematical modeling as well. So if you're into coding, if you're into math, this could be a lab that you could join. And also, he actually told me last week that he's looking for an undergrad to join the lab. So email him if you're interested. Yeah. Okay, next slide. So I'm also working in Dr. Bubb's lab, and I actually got connected to Dr. Bubb through Dr. Schreier. So that's something cool. He's a collaborator. So Bob's lab focuses on the dynamics of excitable cell networks. So he's more like computational, more imaging, and he uses these tools to study complex cardiac dynamics for more like arrhythmia research. So yeah. So first, I started as a volunteer in Dr. Schreier's lab. So in October of 2023, I reached out to Dr. Schreier. I'll talk more about how I reached out to him later in my presentation. But by December, I finalized my role in his lab, and in January, I started volunteering 3 to 4 times a week. So first, I had to get a lab certification training, which I'm sure most of you guys will also have to do. It's through McGill, so I think it's through my work day or something. And then after that, you do like I learned a lot of wet lab techniques, so like cell culturing, Initially, I was just shadowing the PhD student who was working in the lab. So I did a lot of just like observing, taking notes. And then after a month or two, he was like, okay, you can take charge and you can start actually doing these yourself. Also, at the same time, I learned about lab maintenance, so how to clean, the different types of waste, et cetera, et cetera. And then come the summer, I became a paid undergraduate research trainee. So initially, the goal for me in the summer was to thaw pluripotent stem cells and then differentiate them into cardiomyocytes. But I ended up being able to do more than that. So I did the culturing and differentiation, and then I was able to actually form spheroids. If you guys have any questions about like my research, feel free to ask me more, but I'm just gonna go through it a little quickly. But yeah, so I formed spheroids, and then using these spheroids, I tried to paste them optogenetically using Dr. Bubb's blue light system. So here you can see in the diagram, it's like a non-excitable cell. It's like a hex cell. I'm not sure how many people are familiar with hex cells, but they're transfected with a light-sensitive channel. So if you stimulate them with light, you'll be able to stimulate the cardiomyocyte and control their beating rate. And this is really applicable to arrhythmia research and like pacemakers, etc. And then at the same time, I also did some coding for Dr. Bub. So I had some background in coding in Comp 204, which is a prereq for physiology. And then I also took Comp 250. So with Dr. Bub, I did HTML and Java coding for one of his codes because he's working on this pedagogy program for use in Fiji 210 and Bio 309, which is mathematical models in biology. So I worked on some code with him as well. And then now I'm currently conducting a research project under Dr. Bob. So he's like my official supervisor with collaboration from Dr. Schreier. So my goal is to measure the phase resetting properties of cardiac spheroids using optogenetics. So essentially what I'm doing is I'm pairing the cardiomyocyte spheroids with the hex spheroids transfected with channel rhodopsin. And then I'm going to paste them optogenetically and see their phase resetting curves. And essentially, a phase resetting curve is just how external stimuli can affect the beat rate of a cardiomyocyte. So the implication of this is to understand the complex cardiac dynamics and biological oscillators, and this is really relevant to clinical research in arrhythmia. Next slide. You're good.
Sometimes it doesn't. Oh,
wait. Why did it go back?
yeah, I don't know, you know. Why it is wrong.
No, you're good, you're good.
Okay.
Yes. Okay, so basically I just talked about my plan, so I'm not going to go over it again. But currently I'm just working on spheroid formation and optogenetic stimulation. So I had some like setbacks in terms of like the progress and just like difficulties, but you know, that's a part of research. So yeah, I just gotta troubleshoot and yeah. Okay, next slide. So now I'm just going to talk about some research tips for you guys. So in terms of choosing a lab, I would say like don't just choose based on like the research focus itself. You have to also, or you should not have to, but you should also familiarize yourself with the environment of the lab. So for me, for example, I was able to tour some labs and really just get to see the dynamics and like how like who the people were, how they work together, and like the vibes kind of sort of thing. So also evaluate the environment because that's going to be a huge part of your experience. And then also cold emailing is perfect. It's very good. But also I would recommend speaking to them in person. So for me, for example, for Dr. Schreier, I was taking his Fiji 209 class. And then it was like this one day I went into his office just like unannounced and he thought I had a question about the class. But then I was like, oh, actually, I'm looking for a research opportunity. And we started talking. And then he was like, yeah, sure. Like you're welcome to join. So that was, yeah, I'd say talking in person to them is much more effective because they remember your face. And then also, if you want to use the cold email template that I used, or if you're interested in seeing, like just send me an email or a text and I'll send it over. So once you're in the lab, I'd say take the initiative to find the opportunities. Like they're not just going to come to you. You have to actually like take the initiative to look for opportunities, ask for opportunities. For example, for me, with Dr. Bub, like if I didn't reach out to him, like I never would have been able to code. It was more just I was kind of like looking and seeing what projects were going around like in the lab, and then I saw that project and I was interested, so I reached out to Dr. Bub, and that's how I became a part of both of their labs because initially I was just with Dr. Shire. But yeah. And then also I'd say read up on relevant papers and use to really understand your field more and to be able to conduct relevant experiments. Um, Also, make your expectations very clear because for me, at least at the start, I didn't really make my expectations clear in terms of how many hours I was expected to work. So I ended up working a lot more than I would have liked like the first month. But then I talked to the profs, I talked to my PhD supervisor, and we all got it sorted out. So yes, make your expectations clear at the very get-go. And then also expect to make mistakes and have a lot of setbacks. At first, I made some clumsy mistakes, just like stupid mistakes, but you know, you learn from every single mistake. So, yeah. And then also, I'd say go to the lab events and socials. There are a lot of like things that go on. So there's like dinners, there's like wine and cheese events that happen. A lot of like graduate student things that like the undergrads get invited to if you're a part of a lab. So I'd say go to those because you get to meet a lot of cool people and different professors in different fields. And yeah, and also it's okay to switch labs. I personally have never switched labs, but I know a lot of friends who have, and it's totally okay. The profs will have nothing against you. So if the lab is not for you, then totally just it's okay to switch. Yeah. And that is it from me.
Yay. Thanks, Lorena.
Okay,
Okay.
so I'm Alexa. I'm in U3 Physiology and Math at McGill. So I'm currently working in Dr. Cook and Dr. Krishnaswamy's lab. They have like a joint lab together in the McIntyre Medical Building. So the area of research is neuroscience, and we're focusing specifically on visual attention in mice. Just to give you a bit of background information on what it is. So we use mice as our model, and they're actually genetically modified for the neurons in their brain to light up when active. It's actually pretty cool. At first, when I heard that, I was like, wait, what? But yeah, so the top part of their skulls are actually removed surgically, and they're replaced by a clear window so that we could see inside their brain and like get some brain images. Or they can even be genetically modified to have clear skulls to begin with. So that allows us to see their brain and look for signals during the task that I'll tell you about in the next slide. Not yet. And so, also for the task to be successful, there needs to be some kind of incentive for the mice. So the mice are actually water deprived, and they receive water by successfully completing the task. So I know it's a little sad, but that's just how it works. And I'll show you an image of what it kind of looks like on a mock mouse skull, because basically they need to get a head plate put on their head so that their head could be secured during the task to be able to take those brain images. So yeah, next slide. So this is actually a real mouse skull, and Dr. Cook asked me to basically get one, like order one, find one, to be able to um, basically use it in the experimental setup and kind of move the cameras around because we have cameras for the to take videos of the eyes. So my task was to find a mouse skull, and I made those little eyes out of like putty, and we glued the head plate on. So that's kind of what it looks like. And so that head plate, it's kind of glued to the skull of the mice. Yeah. So as for the task, the concept is that the mice undergo gradual training, so over like a few weeks, for them to be able to detect gratings through the dynamic noise. So it's kind of like black and white squares on the screen, and through those squares, there's like a 3-bar or vertical grading that you could see in panel A. And so the by training them, like the task gets harder and harder as the weeks go on, and the mice actually perform pretty well and can detect gratings when there's a lot of noise and they're kind of hard to see. So that's actually pretty interesting. And then you can basically by making it difficult, the mice have to pay attention. And then you can look for some kind of brain activity that can tell you that they are paying attention. But it's a very like complex thing, and I don't even fully understand what methods they use to actually detect that they're paying attention. It's a very kind of abstract concept, but yeah, so that's the idea. So yeah, in panel A, you can see that they have two screens, one on each side of their face, and they each have kind of dynamic noise on the screen, and only one side will have the grading appear. And they do like many trials, and they also do like blocks. So each block will have the grading on a specific side. So like they'll do, let's say, 30 trials on the left side, and then they'll switch to the right side and do 30 trials there so that they can record for both kind of sides, both eyes. And so panel B just shows kind of how they set up like the noise plus the 3-bar grading. And like you'll see on panel D, they have something called coherence. So I think it's that the higher the coherence is, the easier it is to see, and the lower, or it could be the other way around, but that's the basic concept that they use. And so it gets harder and harder as the task goes on. Yeah. So basically, the task starts when the mouse hears a static cue. So it's a noise coming from one screen. So the screen that's going to have the grading appear. So that's to tell the mice to start paying attention to that specific side. And so the mice get trained to know that they need to lick the spout in front of them to start the task. And then if they lick in the time period between the start lick and the grading appearance, that's called a false alarm lick, and they need to restart the queue and restart the trial. And then from there, if they wait long enough and they're like patient enough, then once they see the grading, they lick, and if they lick like in the time period where when the grading is showing, then it's called a hit lick. And if they, let's say, lick a little bit too past that point, then it's going to be a miss lick. So that's how they kind of quantify the licks. Yeah. And so, yeah, to make sure that, let's say, the mice aren't like estimating the time between the start lick and the grading, they vary the time between, well, basically that time period. And they think that the mice use one of two strategies. It really depends on the mouse, actually. It's pretty funny. So they can either like try and estimate when the grading is going to come on, like from their previous experience, or they look at the contrast on the screen, like the contrast itself, to detect the grading. And that can explain the false alarm licks because let's say there's the static noise kind of looks like a 3-bar grading because of how the little black and white squares are aligned. Like the mice might use that and lick when it's actually not the grading. Um, so yeah. And so what I've been doing for the lab, during the summer, I volunteered about twice a week, and I was doing more like coding stuff. So I was using a software called FaceMap to get information on the mouse eyes. So like the pupil area, the position of the center of the pupil. And so this could be analyzed in conjunction with the brain images and kind of synced together. And then you can see if, like, let's say the pupil dilates at the same time as like a brain activity happens, like that might be an attentional signal. But yeah, so that's not done yet. but we're just in the process of kind of analyzing videos using FaceMap. And yeah, so it started with the volunteering, and then in the fall, I decided to do a 396 project with Dr. Cook. And so just kind of continued with that concept of like analyzing the eye data and making sure that FaceMap is accurately kind of analyzing the data and finding the pupil. So, because that's what FaceMap has to do. It has to kind of, we give it images and it finds the pupil and gives us an output of data. So you could go to the next slide. I have just an example of that. So this is the software. So I basically have to set a few parameters and then it'll kind of find the pupil for me. And then I had to kind of replot things in MATLAB, like re-upload those videos and plot what FaceMap outputted to me to make sure that FaceMap was accurately doing it so that we could keep using it. So that'll be in the next slide. So yeah, here you see the little red circle is the pupil, obviously. And then the kind of, I don't know how you call this, crosshair or something, that's showing the center of mass. So by that image in the right corner, you can see that FaceMap is properly identifying the pupil. Uh, And so, yeah, so that was kind of the later part of my fall project. And it was very different, kind of took a different spin than the rest of it, which was coding. But it really just shows that like it depends on what the professor like wants from you and like needs from you at the moment. It definitely helps that I had coding experience and that I was in the program that I'm in, for him to have chosen me because he knew that like I could help him out with some analysis stuff. But yeah, it was also interesting to explore this aspect of it. Um, and yeah, so just a few benefits of joining the lab. So I was invited to the weekly lab meetings, and that was pretty cool because it would either be grad students presenting like a paper related to their research, and then sometimes like the professors would criticize that paper even and like say it wasn't really that didn't hold up too much or something. So that was interesting. Or the grad students would present their work or their figures that they were going to like publish in a paper or in their thesis. Sometimes the professors would even present what they were working on. Some of the things I didn't understand, but it was cool to even just like get into that environment and to hear kind of what they had to say and like what their progress was on, even if I didn't fully grasp it. And yeah, just to see the lab environment and the day-to-day life of a grad student if you're thinking of applying to grad school, as well as getting a letter of recommendation from the professor to be able to apply to grad school because that really stands out. And yeah, it was just cool to see science in action.
Mhm. Awesome. Thanks so much, Alexa. That was super cool. So that closes the part where we're talking about our own research. So now I'm just going to mention two fun events happening with the McGill Scientific Writing Initiatives, and then we'll open up the floor for questions. So if you guys have any writing assignments coming up or lab reports, definitely come to our writing session next, like this upcoming Monday. It's going to be Red Path Pod 4 we reserved, and our alumni advisor Sydney, who's a PhD student right now in nursing, is really well educated in research writing. She's been doing a lot of it. So she'll be there to answer questions, have a guided writing session. You guys can all just write together for 2 hours, come whenever you want, or stay the full time. And she'll be there to answer questions and give a lot of amazing tips. And then we also have our annual science communication competition. So this is a really great opportunity to actually get research experience. Part of this competition is writing a literature review, which is really a big, big thing in research. It's where you compile a bunch of information from a bunch of different papers and make your own research paper. The winner of our competition gets an opportunity to get published in the McGill Science Journal, which is a peer-reviewed journal. So you go straight to the peer review process, and then if they approve, then you're in their journal. So that's really cool as well for your CV, for your growth as a science student. And together with this competition, so you have the literature review, but before that, you have two other tasks that you complete on your own or with a team. And this is a creative task where you express a topic in science, let's say Alzheimer's, the immune system, um, and you can write a picture book we had one year, or a podcast, an interview, and this is just a way to get, comfortable in science and writing science. And it's a really fun opportunity. So definitely check it out. You can scan this QR code for more information and to even apply. It's starting after winter break. So yeah, before questions, just to let you guys know, you can send us your email in the chat or DM us on Instagram with your email because a lot of people in MSWI are in grad school and come across a lot of opportunities for undergrad students. So if we see anything, we can email you and you can choose if you want to apply for it. And yeah, so that could definitely be, helpful for you to find, you don't have to look out for an opportunity. We'll give you one right away if we find one. So definitely send us your email to be in the loop with that. But other than that, yeah, do you guys have any questions? You can say it in out loud or in the chat. You could direct it to one of us or just overall questions. Oh yeah, thanks for your email. Yeah, you guys can all send your email. Yeah, awesome. Yeah, if you guys don't have any, oh, mhm. Oh, I guess Lorena, you can answer that question.
Um, I would say don't ask to get paid first. I'd say your initial email, just don't mention money, like in general. Um, only honestly, I'd recommend just not mentioning money. And like, because especially if you don't have prior experience in a lab, I think it's generally better if you just start as a volunteer and just like show your passion for actually just like being in the lab itself rather than like being more like, So I'd say don't mention money.
Yeah. But also if you really don't want to volunteer at all, and then like you wouldn't even accept a volunteering position and you'd only accept working, then that's probably fine, right? Like it's only if it depends what your intentions are.
Yeah.
Or you
I
even
guess,
think
yeah,
like
it depends on
people
your
like
intentions. Like,
they
yeah, it's like if,
discard.
But aren't there also
yeah.
like research like awards that you can get? So you can maybe
Mhm.
apply for that and then come into the lab saying like I don't know how
Yeah.
that works. And like if you can possibly get it before you even reach out to a professor, I don't know if like
Um,
timing
I
on
know
that
one
is like
example is the
yeah,
global
there's something
health.
called NSERC.
Oh yeah,
insert.
that too.
Hmm.
Yeah, there's
Yeah,
MSWI
there's
and
like this.
global. Yeah, yeah. No, go ahead.
Sorry, I'm just gonna attach the
Okay,
link for
yeah,
NSERC
catch
in
that.
the
But I know
chat.
for global
Yeah.
health, they didn't open the applications yet, but I applied for that last year, but I didn't get accepted. But I know someone who got accepted the second time that they applied because it's so competitive. So don't get your, don't get discouraged if you don't get accepted right away. But if you do apply, you apply to get funds, and it's directly with a supervisor. So there's a list of supervisors who are partnered with the Global Health Award. And then if you get accepted, then you get funds and you get paid. And then you already have an opportunity. And this is for the summertime. And then yeah, when is a good time to start looking for summer research positions? So I started a bit before winter break. Um, but
Yeah. That's a good time to start.
Yeah.
Mhm.
So like right now.
Yeah, right now. Yeah, if
Yeah.
you're free right now, do it right now because exams are gonna pile up, so you might as well. I remember doing it about now, and then I met with a potential PI like right before exams.
I think I did it more in February, I feel
Yeah,
like. I feel like I did it winter semester, but like beginning winter semester. Um, yeah, but I followed what Ellie uh took from the
yeah,
conference
email.
well, yeah, from the workshop that she went to. So the emailing at least
Like did
five
it
professors
today.
at first is a good thing. Like only one of them answered. I think two of them answered. One of them said he's looking for full time, and then the other one was Dr. Cook. So
Yeah.
that worked out. But yeah.
Yeah. Yeah, and then someone else asked any tips on summer research award applications. So I know for one, finding them, they didn't send it out yet, but I remember last year they sent out emails to your Outlook. So you don't have to go scavenging. Like they literally send you these, this is an opportunity, this is an opportunity. So I remember getting a bunch of emails about that, about NSERT and about the global health. Um, Another option is like searching it up. In I'm trying to think where exactly, but I could definitely when I find it, I could send it to your email since you put it there. But I remember online there's like a site. I don't know, Lorena or Alexa, if you've seen this, a site with like a bunch of volunteer no, sorry, research awards. But I
Yeah.
could look for it.
Um,
I'm not sure if
and also, sorry, in terms of like actually applying, like if you want your application to stand out, I found this like I think it's an interview, but it's basically just like tips on applying for an NSERC scholarship or fellowship. So I can like attach that in the chat for you guys. But also it's like not like sometimes it's like if you start working in a lab like before the summer, like they'll automatic like the prof will automatically just like help you apply for a summer like award. So honestly, it's like if you start before the summer, like the prof will help you a lot. So that's like a benefit of, I guess, starting in the winter semester.
but does it, I think it
But
might depend on the prof. Because, like, let's say for me,
yeah, it does.
he
It
kind of
does.
said, like, he basically said, like, I won't be paying you. Like, this is a volunteering position.
Oh,
But it
okay.
worked out because I could only do like part-time. So that was okay with me. But, like, I think you just have to make sure, like, that yeah, what the professor's thinking for that.
Yeah, yeah, it's different for every lab, for sure.
Yeah. And yeah, for like the looking for summer research awards, I just searched up online like McGill Undergrad Research Award and I found like all the ones that they're gonna email us about in the coming months, like Sura and CERT. So they're all like online, pretty easy to find if you're looking for them, like, but they'll also send us an email soon. And okay, yeah, and then for the next question, what advice could you give if you feel like you wouldn't be up to the task, like if you're missing prior knowledge or techniques? Probably Alexa, Lorena, you guys would be better at answering that. I know you guys had complex stuff to do.
Um, what type of lab do you want to join? Is it more like wet lab?
I think like a lot can, oh,
Okay. Okay. Well, in terms of wet lab, I mean, like for me at least, I also had zero experience before joining the lab. You just have to make it clear to them that you have had no experience before, and then like they'll start slow with you. Like honestly, it's like for me at least at the beginning, it was also very intimidating because I was like reading on the research and the techniques are very advanced, but they definitely like you start like at the very beginning and they teach you like one by one, so it's not overwhelming at all, and you just have to like make your like expectations clear to your prof, like what you know or like what you want to learn, like what you don't know, just so you can like
mm,
yeah, you'll be fine. You'll be fine. Speaking from experience.
Yeah, they definitely like if you don't know what it is, if you don't know how to do it, if you just tell them just point blank, like, can you help me with this? I know in my lab, they explain a task to me and then I start doing it and then I forget something that they told me or I run into something I'm not sure about and they're usually like other students or the research coordinator is like right there, right by beside me. So I just be, I mean, can I ask you a question? And then they really have no issue answering me. And I do that pretty often. So yeah, don't be shy. You'll see. You can definitely get a feel of who you're working with, and you could ask them, what's your question and answer style? Do you want me to ask questions throughout or keep them all, you know, at one time period during my volunteering or my work? So yeah, you can
Yeah,
definitely play it by ear. I wouldn't worry about that either.
I think they also like realize that, any undergrad, this is their first time like stepping into this kind of field. So they don't expect you to already have all of this knowledge, like unless they feel that you need certain courses to be able to understand what you're gonna do. Like most of the time, like they're patient with you and like you go at your own pace and like they help you through it normally. Yeah.
Mhm.
No coding
Oh yeah.
experience. Um, no, because most wet labs actually don't require any coding background. Um, for wet labs at least, like it's more just like actual experimentation. So you don't need coding experience.
Okay, I guess any last call for questions before we end the Zoom? You could always reach out to Miguel SWI by DM on Instagram, and then you can direct the question to any of us, and then we'll answer. So feel free to do that. I'm gonna end the recording.